Let's look a bit more at the physiology of skeletal muscle cells. Now, the nervous system is primarily what controls these muscle contractions. We're really talking about skeletal muscle in this chapter. We'll look a lot more at cardiac and smooth than others. When you want to tell a skeletal muscle to contract, largely that starts with the brain, the motor cortex and the cerebrum, which you'll see in the brain chapter if you haven't already. So when you want one of these skeletal muscles to contract, primarily starts with a group of neurons controlling these individual muscle cells. Now, where all this starts is with something called the resting membrane potentials. Now, anytime you look at the cell membrane of a neuron or a muscle cell, you always talk about these resting membrane potentials. And think about what these are. When you talk about membrane potentials, you're talking about charges on a cell membrane. And where are those charges when a cell is at rest? The charges will be positive on the outside and negative on the inside. That's what that resting membrane potential is. Now, where do these charges come from? These charges come largely from ions. Remember, ions are just charged particles. When you talk about some of the very abundant and common charged particles in your body, sodium, calcium, and potassium are three of the very important ones. Your cells like to keep most of the sodium and calcium on the outside of the cell and most of the potassium on the inside. There's also lots of proteins and phosphates inside the cell, and those have a negative charge. So look at what the cell does. It just simply puts more of the positive things on the outside and more of the negative things on the inside. And that's how the outside charge got positive and the inside got negative. There's your resting membrane potential. And if you look at what sets up this resting membrane potential more than anything, it's these proteins in the cell membrane called the sodium-potassium exchange pump. Every time one of those proteins burns up an ATP, it pumps 3 sodium out of the cell and 2 potassium in. And as that potassium builds up on the inside, there's actually some ion channels that'll let it diffuse back out and add to some of that outside positive charge. But point being, you want neurons or muscle cells to work correctly, you've got to have this resting membrane potential. Now, looking back at these ion channels, remember what these are: proteins primarily found in the surface of the cell membranes. And again, you get to muscles or neurons, there's a large number of them. There's 3 main big types of ion channels. One are called non-gated, also called leak channels. These proteins don't have gates on them, so whatever ion it is that they're particular to can pass anytime it wants. And that material tends to pass from an area of high to low concentration. You saw that with diffusion back in chapter 3. But there's also a couple of types of ion channels which do have gates. These gates can close and not let the particular ion that they're particular to pass, or they can open. Now look at what will open or close these gated channels: a ligand or a voltage gated channel. Now, a ligand is a type of chemical signal. You look at many different types of chemical signals when you get to hormones and such primarily. But what opens or closes a ligand gated ion channel is a chemical signal. There are many neurotransmitters and many hormones which do this. But there's also voltage gated channels. These will open or close in response to a change in voltage. So look at what you've got. Primarily, when cells want to communicate, they're going to do it in one of 2 ways, either with chemical or electric signals right there. Your endocrine system, which is all about hormones, is really good at using chemical signals, ligands. But a lot of that goes on in the nervous system too. Nervous system likes to use the chemical and the electric signals. So a lot of ion channel gates opening and closing throughout many cells of the body at any one time. And remember, each one of these ion channels is specific to one ion. Like a particular type of protein ion channel will let just sodium go through it, or just calcium, or just potassium. Now let's look at something related to those ion channels called action potentials. Now remember, the cell is going to set up at the resting state a positive charge on the outside of it and a negative charge on the inside. To depolarize one of those cells means to swap those charges. In other words, bring the positive charge in and the negative charge out. This is going to happen by moving those different ions. When you depolarize these cells, action potentials, which are electric signals, are generated. Whenever you repolarize a cell, that's when you put the charges right back where they started. Again, there's only two charges, positive and negative. Either the positive's on the outside or the inside, one or the other, and the same goes for the negative. So depolarization, bringing the positive in, repolarization, putting it right back out. They can only be in one of those two places. And when you look at how these electric signals called action potentials work, you'll see that one principle that applies to them is the all or none. In other words, you either get one or you don't. So when something like a neuron or a muscle cell generates an action potential, they're always the same. They're always of the same strength and intensity. It can't generate a weak one or a strong one or anywhere in between. It's all or none. It's all or nothing. You'll see that with other things throughout these chapters too. And when an action potential moves down something like an axon or a muscle cell, those talk about how it propagates. It moves. And one action potential doesn't actually move the length of something like an axon or the cell membrane of a muscle. You actually generate one action potential after another. We'll look at that a little bit further along. And the frequency of these definitely affects how muscles work. But let's look back at these gated ion channels in the action potential. Again, you like to start off at the resting membrane potential. That's where the positive charge is on the outside and the negative charge is on the inside of the cell. Remember, this comes primarily from the concentration of three ions: sodium, calcium, and potassium. The cell likes to keep most of the sodium and calcium on the outside and the potassium on the inside. The inside of the cell contains many molecules, which makes it negative on the inside. Most proteins are negative, and phosphates are too. Now again, if you depolarize a cell, think about what you did. You just swapped the charges on the cell membrane. You just brought the positive charge in and the negative goes out. That's often accomplished in one of three ways. Number one, opening sodium channels and letting the sodium in. Open the calcium channels and letting the calcium in, or closing the potassium and trapping it in. Look at what you're doing in all three cases. You're putting more of the positive ion inside the cell. That's why the inside charge swaps to the positive. Now again, if you want to repolarize this cell, you're just going to return the charges right back where they started. That's generally accomplished by doing exactly the opposite of what we saw in number 2. Close the sodium channels and the cell will pump it right back out. Close those calcium channels and the cell will pump it out. And open the potassium and it will diffuse out, just simply moving from a high to low concentration. And looking again at this action potential propagation, imagine what we've got right here is a skeletal muscle cell or an axon, whatever that may be. Here, right over here on the top left of this picture, and also right below it, we can see where there's a depolarization. Again, when you depolarize this cell membrane, moving the positive charge in, there's where you generate an action potential. Well, when you generate this electric signal here, that'll open up the voltage-gated channels beside it. Positive ion comes in, another action potential is generated. Opens up the gates beside it. Positive ion comes in, generates another electric signal, and over and over and over. So when they talk about action potential propagation, the movement of these electric signals down the length of something like an axon or a muscle cell, notice that one electric signal is not traveling the distance. It just generates another electric signal beside it, another one beside it, and so on down the line. And that's what's meant by the propagation.